The reason why we're here today is to kind of go over a little bit on the changes that are happening with ISO 10993. Right now is a very dynamic and exciting and sometimes challenging biocompatibility landscape. If you've done biocompatibility recently, you might have submitted some things to regulatory agencies and might have got different feedback depending on the testing you did and some of the changes that we're seeing. And in fact, in my whole time on the ISO committee, which I've been on there for almost 10 years now, we have never seen as much development in the 10993 standards as we have in the last 2 years. But today we're going to talk about two categories that are changing pretty dynamically. And they're changing based on a lot of feedback from new science techniques and from different regulatory agency impact. The first one we're going to be talking about is genotoxicity. And then we're also going to be talking about hemocompatibility. That's ISO 10993-3 and ISO 10993-4. Both of these standards are either being revised right now or are finalizing the revisions and going to be taking effect soon. So like I mentioned, the first one we're going to be talking about is genotoxicity. Genotoxicity is very necessary. It's a precursor to cancer. If you have genotoxicity, if your sample has something on it or if there's some kind of leachable that's coming off your device that impacts the genes, then those impacts going to be propagated from cell to cell, and then sooner or later you have potential to cause cancer. And nobody wants to have their device cause cancer. So genotoxicity is very important. It also helps with your justification of carcinogenicity. Okay? So if you want to justify out of your carcinogenicity testing, genotoxicity is very helpful. Now, when we're looking at genotoxicity, we have three layers we have to get down to. Okay? We have the DNA, we have gene, and we have chromosomal. All three layers are important to examine when you're doing a full genotoxicity testing suite. And we have 4 tests in the ISO that we can use to evaluate those three categories. We have the AIMS, okay, we have the mouse lymphoma, chromosome aberration, and then we have the only animal test, which is the mouse micronucleus. So we have 3 in vitro or lab bench tests, and then we have that 1 animal test that can evaluate genotoxicity. So by looking at these different tests, we can kind of get those 3 distinct layers that we need to determine the safety of your device. I'm going to go a little bit through 2 of these tests in genotoxicity, okay? I'm going to touch on AIMS, and I'm going to touch on the chromosome aberration. I'll also kind of mention a little bit of the mouse micronucleus so you can get a background on how the tests are run. The first is AIMS. AIMS is also a very unique test to biocompatibility. Usually we don't like bacteria in biocomp, especially in the cells test. We like to keep bacteria away from our cells, right? This is the only biocompatibility test that we actually utilize bacteria to determine safety. Okay? So we use E. coli and we use Salmonella, and we use specific strains of these bacteria that are engineered in a way to be sensitive to possible mutagens. So if there's anything on your device that can cause a mutation, then these bacteria are sensitive to that. And what that means is they can mutate. Okay, everyone's seen X-Men, they know what mutations are, right? These bacteria don't fly or have X-ray vision, but what they do when they mutate is they change in a certain way. So without the mutation, the bacteria is histidine dependent. Histidine is an amino acid, and these bacteria need them to grow. Okay, so that's what histidine dependent means. These bacteria need the histidine to grow. So if we don't give them histidine, we should not see any growth. Right? But if they mutate, then that mutation allows these bacteria then to grow. Okay? So we take the bacteria, we extract your sample, we put it in contact with the bacteria, and then if there's any possible mutation from your device, these bacteria can grow without histidine. Then it's a simple plate count. All we have to do is count how much bacteria is on your test plate compared to the control plate. Okay? So if you have a 2-fold increase in bacteria growth compared to your controls, then you're considered a possible mutagen. So that's the AIMS test in a nutshell. So we use these very sensitive bacteria to look for your potential. Now, some of these bacteria are going to mutate automatically, so that's where we run the controls. We want to see if they're natural mutation, and if you have that 2 times more increase than that, then that's when your test sample becomes a possible mutagen. Okay, the next one we're going to talk about is chromosome aberration. Chromosome aberration is done using Chinese hamster ovary cells. Okay? These cells, we grow them up on these flasks, we take them, and then we put these cells on the bottom of this plate. And then we take an extract from your device, and we put them in contact with these cells. Okay? Now, we have the extract on top of the cells, and the cells are growing and dividing. And if there's any possible problem from your device, these cells are now bringing them in, right? Then we stop the cells. We don't let them grow anymore. We freeze them, and then we kind of smush them apart. Okay? And what we're looking for is something called a karyotype. And this is a karyotype. Okay? What it is, it's the chromosomes. So these purple spaghetti-looking things are the chromosomes inside these Chinese hamster ovary cells. And what we're looking for is aberrations. Some of the things, the possible aberrations, we're looking for breaks or gaps or insertions. The one that's very evident here is this quad-looking thing right here. That's quadrihedral. And it's two normal-looking chromosomes have broken apart and formed with two other ones to form it. So it should be looking like this one right here, but it's formed this four-star-looking one. So that's an aberration. So now that we have the cells broken apart and we're looking at the chromosomes, we actually sit there and count how many aberrations that your test cells have compared to the control cells. Okay? So we have someone underneath the microscope sitting there just counting the aberrations. Okay? A lot of counting microscope work. But then, after we get the counts and the types of aberrations, we compare those counts to the negative controls. And if you have statistically higher number of aberrations compared to the controls, then once again, you're considered a possible mutagen. Okay? So that's chromosome aberration. Now, the last one I'm going to talk about in genotoxicity is the animal test, the mouse micronucleus. And this is kind of an umbrella test. Okay? So this is using an animal model to try to get to the actual metabolism of your extract or your test sample to see if we can see anything pass on to the mice. Okay? So it kind of encompasses all those three aspects I talked about, the genes, the chromosomes, and the DNA. Okay? So what we do is we have 45 male and 45 female mice, and we give them interperitoneal injections, which means we inject your extracts right here in their body cavity. Okay? So we inject the mice with your extract of your device, and then at 24 hours and at 48 hours, at 24 hours we take 5 animals, and at 48 hours we take the rest of them, and we harvest the bone marrow from the animal. Okay? And what we're looking for is something called micronucleated polychromatic erythrocytes. That's kind of a long thing to say, and us scientists don't like to say long things, so we shorten it down to PCEs. Okay, so these PCEs are signs that something has mutated in these mice. Okay? So the more PCEs we're able to examine in the bone marrow, the more mutation that's occurred in this mouse. So once again, we statistically measure the amount of, or number of PCEs compared to the controls. The higher the PCE count of your test, if it's statistically higher, then once again, you have a possible mutation. Now we get into the regulatory impact of genotoxicity, probably the big reason why you're here. Right now we're going to be comparing two testing strategies. ISO 10993-3, this is the current standard as it sits today, and the FDA recommendation. Okay? So right now the ISO 10993-3 gives us two options. Okay? Option 1, in fact, these are switched, so I apologize. Option 1 is Ames, mouse lymphoma, and chromosome aberration. Okay? So we didn't talk about the mouse lymphoma, but it's another in vitro test. But basically, you need to encompass those three areas using these three tests. Then option 2 is Ames and mouse lymphoma, and then we can size the mouse lymphoma colonies. The concept there is that the size of the mouse lymphoma colonies should give you some kind of chromosomal damage information. But the problem is right now we have one option that has only 2 tests, and we have a second option that has 3 tests. So which one was most medical device manufacturers using? Which one would you use? 2 tests or 3 tests? Two tests, right? Cheaper, quicker. Okay, so the FDA reviewers were getting this test that had Ames and mouse lymphoma and then had sizing of the mouse lymphoma colonies, and they were going, okay, sizing's supposed to give us some kind of information, so they were calling the labs, or writing back, not calling, but writing back the labs and saying, what size determines what damage? Where's the cutoff point? And no one in the industry had a good size determination. When is a small and when's a large? So most of us were just going, well, we had this much size, you guys determine what it is. And so there wasn't this good consensus information as far as the size of the mouse lymphoma. So the FDA released its own guidance document, okay? And right here is the FDA website. I'll make sure that the presentation is on the website, on the Canon, so you guys don't have to write down that. So this is a lot easier, so you can just have it. But that's the guidance document. But what it says is it says, okay, we want you to do an Ames, but then you get to choose either the mouse lymphoma or the chromosome aberration. So you choose between those two tests, and then you have to do the mouse micronucleus. Okay? If you remember, the mouse micronucleus was the animal test, the umbrella test. And the ISO 10993, the two option, doesn't even list the mouse micronucleus. That's because in the later text, it's used as a confirmation test. If you got a positive in one of these in vitro tests, then you would do the mouse micronucleus as a confirmation of that positive. Well, the FDA says, okay, we want you to do the Ames and that animal test, and then either you do the mouse lymphoma or the chromosome aberration. So this is one instance where the FDA and the ISO 10993 do differ a little bit. Now, the only guidance I can give you between the mouse lymphoma and chromosome aberration, they're about the same cost, they're about the same turnaround time. So the only benefit is the chromosome aberration is more highly accepted in Japan. The Japanese Ministry of Health loves the chromosome aberration. And in fact, they tried pushing the chromosome aberration and Ames test only at the ISO 10993 meetings in Berlin. It didn't quite stick, but they believed they had good data to demonstrate that an Ames and a chromosome aberration can pick up all the possible mutations on a device. So they really like that chromosome aberration. So if you're ever going to market your device in Japan, you might want to lean towards the chromosome ab, but really they're interchangeable as far as the FDA goes. So the next category that we're actually changing quite a bit is hemocompatibility. We're still in the middle of changing this standard. So much so that we're going to be meeting in a couple months down in San Diego, and we're probably going to be doing round-robin testing all over again to try to come up with a testing scheme for hemolysis on this. So this is the latest up-to-date information as far as where we're at. Hemocompatibility, in essence, we want to look to see how your device interacts with blood. The most important thing here is we all know that when you have surgery, there's going to be blood present, right? We know, even if you go into like a little pinprick, there's going to be blood. That's not the blood interaction we're talking about, okay? The blood interaction we're talking about here is circulating blood or used blood. You have to have some kind of contact with a circulatory system to be looking at hemocompatibility, okay? Waste blood doesn't matter. So either some kind of absorption through a membrane, indirect, like an IV bag going into the circulatory system, or a stent that's directly embedded there. Those are the things we're looking for as far as hemocompatibility. And these are the lists of the tests in the standard that we're evaluating hemocompatibility. So we have thrombosis, we have complement, hemolysis. Okay, so there's a list of tests that you have to look at. Okay, so this is the big problem right now with ISO 10993-4. This is the table 1 out of the standard. And table 1 is supposed to be used where over here you have an example of devices. So you look at your example and you say, okay, I have a catheter. So here's my catheter. Come over, I need to do thrombosis, coagulation, and hemolysis. This is going to be thrown out the door. In fact, I'm on a committee that we're rewriting this chart. And we're going to be basing it off surface area. Okay? Surface area is much more impactful to how a lot of these tests react. A very small device is not going to impact the blood as much as a big, large device. Okay? So we're going to be basing it off of surface area. The discussion right now is, once again, where's a large and small surface area break off? right? You don't want to be that 1 centimeter squared over that point. So we're in discussion to try to figure out through data what's impactful to the patient. So this chart's going away, and it's going to be based off surface areas in the future. The problem is, though, is right now, for example, the FDA does not accept ISO 10993-4. And specifically, this is the chart they don't accept. So you need to submit today. What testing do you need to do? Us at ISO, we're kind of slow at bringing out standards. We're extremely slow at bringing out standards. And I know that a lot of the regulatory agencies out there actually ask us to be faster because they're desperate for more information. But if you're looking at submitting blood stuff, what do you do today? Well, that's a little hard to say. We don't have a great definition of what testing you need to do. What I can give you is latest trends depending on the regulatory agency you're submitting to, okay? What I'm going to be talking about today are 2 tests that you must do if you're submitting to the FDA. That's hemolysis and complement. Now, if you can see, complement up here is only listed in a couple things. But the FDA feels like there's a lot more risk to the patients than that we've been testing. And it's a big potential risk. Complement, the end result of complement is anaphylactic shock, okay? So the risk to the patient is pretty high. So the FDA has a big concern with complement. So to kind of give you an idea of what we look for hemolysis and complement activation, this is hemolysis. This is kind of the screening test for hemocompatibility. It's quick, it's cheap, and it can kind of give you a good idea of how the blood's going to interact. This is the only test, though, that we've run both an extract and a direct contact. So what that means is we both put your device directly in contact with the blood, and we extract it at the same time. The reason why is because the minute blood leaves your body, it starts to hemolyze, right? And we can't keep your device in contact with the blood for more than 3 hours. So if your device is in contact with the body for less than 3 hours, then you could probably use the direct contact and have a good justification. But for devices that have longer than 3 hour contact, we have to do the extraction method to replicate that long-term exposure, okay? But the direct contact is important because the blood, most of the hemolysis that can actually happen through mechanical means. Okay, rough, sharp edges, or a certain type of material that the red blood cells will burst while hitting the wall of the material. So we have to look at both direct and hemolysis for this test. So this is how it's run. So we either put your device directly into the blood, or we do an extraction and put that extract in the blood. And in this case, Nelson Labs, we use human blood. One is because we're right there and we can bleed. So, and 2, human blood is a little bit, well, that's one thing we're doing the round robin with right now, but a lot of people feel human blood's more predictive than, let's say, rabbit or bovine blood. So we use human blood, we put your device in the blood or extract it, and then we look to see what happens, okay? So we spin, after we have it in contact with the blood, we spin it down. If the red blood cells haven't burst, then they form these pellets on the bottom, okay? So your red blood cells are here, and they're whole. But if they have burst, red blood cells have something called hemoglobin in it, right? And hemoglobin is red. So if they have burst, the solution will be red, even after you spin it down. So all we have to do is do a spectrophotometer to see how much red is in the solution. The more red, the more hemolysis. The less red, the less hemolysis. So we do the same thing with your device. This is negative control. We use polypropylene pellets. For the positive in this test, we actually use nitrile gloves. Who knew, right? So nitrile gloves will give us this positive result. Polypropylene will give us a negative. And we see how your test compares with that. So, and then we base it on this percentage. So 0 to 2, non-hemolytic. 2 to 5%, slightly hemolytic. Anything over 5% is hemolytic. That's based off the negative control. Okay, so the next test is the complement activation. And this is the one test that, like I said, 2 or 3 years ago, we weren't seeing a whole lot of requests. But because of concerns from regulatory agencies, we've seen a big increase in this test. Most devices that have contact with blood have been using this. We have to use human blood. We actually use plasma in this case, in our test. We also have to have a direct exposure, no extract. And the one thing we highly recommend with this is that you run a predicate device with this test. And there might be some other labs out there that don't recommend using a predicate or don't tell you. even if you test with them, please run a predicate. Because the human blood chemistry changes so much from day to day. Okay? It matters what you ate, it matters what you exercise. If you ate that carrot cake right now, tomorrow your blood, it's not going to be too nice. So the blood changes so much that even if you take your same device and test it from day to day, your results could be different. Okay? So what does that mean for pass-fail? The only way we know is to compare it to a device that's already on the market. Okay? So we use a predicate device. We also use controls, but the controls are polypropylene on this end and cobra venom on this end. Okay? Chances are you're going to be between those two, and hopefully closer to polypropylene than cobra venom. So a lot of times we'll run the test. If you don't have a predicate, the results will come back and they'll say, well, statistically you're higher than polypropylene, and good, you're statistically lower than cobra venom. But the regulatory agencies will go, well, what does that mean? You know, tell us what's your complement activation. It looks like you're activating complement. Well, you are activating complement. Everything activates complement. What's important is determining how that relates to safe products on the market. Okay? That's where a predicate device comes in. So we use the plasma, we do a direct extract, and then also you have to look for two proteins, okay? C3a and SC5 beta 9. Those two proteins, and it's not important that you remember the names, just make sure you're looking for two proteins, okay? Classical and alternative pathways are another way to think about it. Both pathways have to be tested, okay? And this is why. This is for those biochemical nerds out there, us geeks band together. So these are the two pathways. We have classical and alternative, and both of those can activate proteins or activate complement system in a different way. So that's why we have to look for both C3a and SC5 beta 9. So kind of the quick sum up on the changes, the -4, the new chart, okay, that's going to be looking on surface area, and hopefully be able to have good guidance on what testing you need to do for your device. Molly Gosh from the FDA is on that committee with me. So she is doing some great input on there, and so we're hoping that the end goal of -4 is to have full acceptance by FDA, Europe, and Japan. The three big regulatory input to the ISO 10993 committees. Okay, so those are the three ones that we have representation from each one of those regulatory agencies on the committee and on this working group that we're part of that's be looking at that. More detailed procedure in the standard. We're actually trying to identify what hemolysis, so for hemocompatibility we have ASTM, we have NIH, and the Japanese method. So those are the three different kinds of methods for hemolysis test. It looks pretty easy, right? I showed you a nice picture, and there's some differences between those three methods though. And those three differences can make a big impact on results. So right now we're actually doing round-robin testing. We're starting round-robin testing where we're going to be looking at all three of those methods, NIH, Japan, and ASTM. And we're also going to be looking at there's a difference between human and animal blood. Okay? And so we want to see what test procedure is the best, what one can be most reproducible between lab to lab, and then we're going to be putting more detail in the standard for those procedures. What this means for you is less variability between labs, and results, okay? So that's what we're going on right now, and that's the last point. So any questions at this point? I think I left about 15 minutes that we can go over any questions you have regarding your device or the testing in general.
I have one.
Yes.
What if you do your predicate along with your new test, and you're doing it for the first time, and your new one ends up closer to venom? Like, is there a defined line that says go, no go? I'm just the nervousness of putting a predicate in. Now you've got something to align it with, and you don't know the answer.
Great question. So her question was, if you couldn't hear, that let's say you do the complement activation test and you have a predicate device, and let's say you run the predicate and your test together, and you're much higher than your predicate, and maybe you're closer to the cobra venom?
Yeah, I mean, just what do you do when you're one-off, or either way, how do you gauge, what's your...
So how the test results are formed, or we statistically compare it to that predicate device. So if the predicate is really high, then you might be fine. You might get a question of, wow, why is your predicate so high? But at least you can say the predicate, as long as you can show your predicate's had a history of good use on the market with no complaints, then you can show that comparison and have a justification. But if you are much higher than your predicate device, then what we'll have to do is an investigation on how to, for a result and a test. Dr. Nelson.
What my job at the company is to go into work and sit in front of a computer and read all day as a science officer. So I read all sorts of really awful stuff, including federal registers and things like that. And the FDA has requested of Congress, and a bill has been introduced into Congress to authorize and empower FDA to reject submissions that use a predicate that they don't like. I don't have it with me, but if you email me or you email Thor, we can give it to you. Now, will it pass? I don't know, but it does reflect the agency's thinking about the selection of predicates. They want to have the authority to say, well, that product is already in a regulatory complaint or having problems. I have a warning letter. We're not going to accept your use of that predicate. So, quoting from the Holy Grail, the search for the Holy Grail, choose wisely what predicate you use, because if you choose a predicate that is going to, and there's a risk here that a competitor's predicate that you choose has a problem after you've started the test, and then you may have, again, issues. So I will get you the bill number, but I'm not sure it will pass. But I wanted you to understand how FDA feels about predicates. They're looking for more authority to be able to reject if you choose the worst of all the predicates on the market. That's another issue that we often see as a laboratory. Say, we'll test 2 predicates and choose, no, you choose the predicate. And you probably can't choose one predicate for one test, another predicate for another. So you need to be standardized in your selection of predicates. At least if this law passes. If it doesn't pass, FDA may do it with a guidance document, which they don't need any regulatory or input.
I get that question all the time. Her question is, what if you have a new device and you don't have a predicate? We always try to find some kind of predicate. Okay? So it doesn't necessarily, well, the best predicate is same material, same contact type, same duration, but very few devices have predicates that meet all those realms. So what we want to look for, most importantly, is that contact type and duration, right? Is that device in the same part of the body and the same duration as your device? Because that's going to give you an impact to the result in the body. The other thing, though, is surface area. It's very important. If your predicate's very, very small, like I talked about before, blood chemistry with surface area interactions, if your predicate's very small and yours is huge, then you can run into a lot of problems. Specifically, we didn't talk about it in this one, but the dog thrombo is very dependent on device size as a result. So it doesn't have to do the same thing as your device does, but it should have the same contact type and duration and a similar size. And if worse comes to worse and we can't find it, these tests, like I said, do have controls. And we can statistically compare them to controls. The problem is polypropylene is a pretty good negative control. So a lot of times your device is going to be higher, and then we have to do some kind of justification of why that is. So it's just easier with the predicate. Did that answer your question?
Our company does a lot of material development and new formulations, and it would be helpful if you guys had any sort of consultants or something to tell us that you can look at our raw ingredients to tell us, yes, looking at that chemistry, you will be okay with biocompatibility testing and some of these tests. I mean, I know you always want to test the end result, but when we have, hundreds of formulations going every which way, do you guys have that?
Great question. We have a couple of things. So her question was that her company makes a bunch of different formulations of raw materials potentially in her device, Is there some way you can go, somewhere you can turn to look at those formulations and give you an idea of the impact of biocompatibility? Is that a good sum up? There are a couple things you can do, and we didn't talk about them on here, but that's okay, because that's what we're doing here. But we do have a lot of experience looking at materials, okay? So if you give us a list of your materials, in fact, you can send it over to me. If you have a list of materials, I can give you a good idea of what we've seen problems with in biocompatibility in the past, either chemically or just material-wise. Specifically, though, the test that we're going to run into the most problems with is cytotoxicity. Okay? We didn't talk about cytotox here, but cytotoxicity is historically the most sensitive test in biocompatibility. In fact, looking through our data at Nelson Labs, if you fail a test, 9 times out of 10, you're going to fail the cytotoxicity test. Okay? Which sounds horrible, but really it's good because the cyto is also the cheapest and quickest test to run. So it's a great way to screen raw materials before you even put them into a final device to see how the impact's going to be to the body. But that being said, everyone's familiar with like USP Class 6 criteria and everything like that. USP Class 6, be careful of USP Class 6. USP Class 6 does not include cytotoxicity. Okay? And as I just mentioned, cytotox is the most sensitive. So I have tons of people say, how can my device fail cytotox? It's USP Class 6. It didn't do cytotox to get USP Class 6. Okay? So a lot of device manufacturers are going to be ISO 10993 compliant. If you can find something that's ISO 10993 compliant, then that means they've done cytotox and probably sensitization, done some more testing to give you an idea. But if you don't know, like I said, you can send a list to me. I'm more than happy to go through it and let you know we've seen that problem with this one before. We've seen it with this one. We have really good database on Nelson of problems we've seen in materials being passed. Did that answer your question? Any other questions? Great. Well, thank you for your time. Feel free to eat more carrot cake. Don't mind my mention about the blood. And thanks for your time.